Hello, biologists. Today we're going to talk about photosynthesis. So photosynthesis is such an incredible process, and it's keeping you alive right now. It might not feel like it. Maybe you don't eat all that many vegetables. Whatever you eat, though, is either made of modified vegetables or you're eating the thing that ate the vegetables. So plants really are the base of the food chain for every living creature on Earth. Here's an overview of how plants function. First, our plant comes in two parts. We have the roots, which is down here, and we have the shoots. And those are legit botany terms, roots and shoots. Each of these shoots is designed to hold up the photosynthetic organs of the plant, and the photosynthetic organ on a plant is the leaves. Now, many plants have modified leaves, so they don't really look like leaves. Like cactus spines are actually modified leaves. And we can talk about that anytime you want. Email me and we can discuss plants. I think I've told you that I have my undergrad in botany. I love plants. I love them so much. Anywhere where you have a large flat place, that's a leaf. That's going to be large and flat because we're trying to increase the surface area of the leaf so that we can get more sunlight on it, because we need sunlight to do photosynthesis. The bottom part of your plant, your shoots, or sorry, your roots, that is designed to anchor the plant, but not only to anchor the plant, to allow the plant to take up water and to take up nutrients that it needs to do its cellular processes. When your plant does photosynthesis, it's maximizing its space, its surface area to pick up the most sunlight. It's also going to take in carbon dioxide, and it's going to release oxygen as a waste product. Here is our chemical formula for photosynthesis: carbon dioxide, water. Then we use sunlight to power the process. This is occurring in the green parts of the plant. Then we're going to make sugar and produce oxygen gas as a byproduct. Let's get into it. The first thing I want to talk about is the anatomy of the organelle, which does photosynthesis. This is called the chloroplast. The chloroplasts are these little green sacs. I like to think about them as shaped like mochi, so that helps me draw it, or maybe a bean, either way. And just like your mitochondria, your plant is going to have two membranes on its chloroplasts. So we have the inner membrane and we have the outer membrane, and the gap between them is also called the inner membrane space. So the vocab for those is the same, which is great. Inside of the inner membrane, you're going to have more space, more fluid, the matrix inside there. That's called the stroma. So that is the space inside. You also have these little flat pita sacs. One of those flat little pita sacs is called a thylakoid, and they stack on top of each other like stacks of coins. A whole stack of them is a granum. These are made of phospholipid bilayer. So again, membrane-bound organelles are so important, only found in eukaryotes. And in this phospholipid bilayer, we're going to have a bunch of proteins and enzymes that's going to do photosynthesis for us. So it's really cool. The space in between the phospholipid bilayer, which is shown in yellow here, that's called the lumen. On your little coin stacks, your granum, sometimes you have connecting bridges. Those are called lamellae. Lamellae means plate. So someone thought it looked like a plate connecting these two things. Why are chloroplasts green? And the answer is chlorophyll. So chlorophyll is a pigment, it's a color. And in your chloroplast, the most abundant chlorophyll is chlorophyll A and chlorophyll B. Chlorophyll A is like more of a deep foresty green, maybe with a little hue of blue. And chlorophyll B is like bright green, like neon green, the green that you see in spring. Both of those are embedded in the chloroplast at just high concentrations. And so when we look at it, we're seeing those pigments. Pigments work because they absorb and reflect different wavelengths of light. So here on the right, I have a graph of relative absorption, and I have the wavelengths of light. This is visible light, light that humans can see. A lower wavelength, the smaller wavelength, shorter wavelength will be blue, and then a higher wavelength will be red. In chlorophyll B, you can see that it absorbs a lot of this blue light, which is really high spiking right there. And then it drops about 500, which is when blue starts to turn to green to our eyes. And it doesn't absorb much in the green-yellow range, but it has another little jump right around the orange-red transition. Chlorophyll A does a similar thing. It absorbs all of the blue, reflects all of the greens and yellows, and absorbs more red. What does that mean for us? That means that when we look at a chloroplast, it appears green because it's absorbing the other colors. So these plants behind me, they are using every color except green. Green is bouncing off of them. I think you can see some of the red pots in there. Those red pots are absorbing every color except for red, and the red is bouncing off of them. That's why they look red to my eyes. Then you have colors like black. So black absorbs all of the color, which is why it feels hotter in the summer. And white, like the door of the closet behind me, that is reflecting all color, absorbing none, because white is a continuation of all of our visible light. So the color that we see as humans is what the item is not using, it's what it's reflecting. So in plants, they're reflecting green and they're using the reds and blues. There are different types of color pigments in plants. It's not just chlorophyll A and B, although most of it is chlorophyll A and B. There is also carotenoids, there's xanthophylls, there's phytoerythrins, there's all sorts of really cool pigments, and each of those will absorb a different color wavelength. And that means that hopefully your plant, if we look here, has a color absorption for every wavelength total, which means that it can utilize everything. You might be asking yourself, why do plants change color in the fall? And the answer is really neat. So majority of the time, plants are showing green because that's what they have in abundance. They have chlorophyll A and chlorophyll B in abundance. Here's the chemical formula of chlorophyll A. Now, when the days get shorter and the winter temperatures start to drop, your plant detects that. It can tell if it responds to stimulus, and it starts to break down chlorophyll. And as it breaks down the green chlorophyll, you can start to see the other colors that were always there, but that were covered up by the amount of green chlorophyll. So when it breaks down the green, you can see the carotenoids and the xanthophylls and the lutein, all of those beautiful colors. Here's beta carotene, those orange, yellows, and reds. And then here is lycopene. Lycopene is what makes tomatoes red. Beta carotene is what makes carrots orange. And lutein is what makes egg yolks kind of yellowy. So each of these pigments is used in photosynthesis, but they're there in a lot lower quantity than the chlorophyll. So you don't see them until the chlorophyll breaks away. So all of those beautiful fall colors are there. They're just hidden the rest of the year. The reason that your leaf is breaking down its photosynthetic pigment and why they will eventually fall off the plant is because in winter, water becomes unavailable. You might be like, but it rains and snows. But as soon as water freezes, it's unavailable to the plant. And in the leaves, there are these little tiny holes called stomata. That's where your plant breathes from. And if water is unavailable and your plant continues to breathe, it loses water when it breathes, just like we do. It will fully dehydrate itself, and it could die of dehydration during the winter. So instead of allowing that to happen, it gets rid of the breathing apparatuses on its body so that it can't dehydrate itself, and it goes into stasis when the water is unavailable. So that's why plants change color when they drop their leaves, and why they drop their leaves altogether. Let's get into photosynthesis. So photosynthesis is going to occur in two places. We're going to have it occur in the thylakoids, which is the sacs of membrane inside the chloroplast, and we're going to have it occur in the stroma, which is the liquid surrounding the chloroplast. This whole round structure here is the chloroplast. The brown is the outer membrane, The yellow is the intermembrane space, and the green is the inner membrane. So this whole process is inside the chloroplast, but in two different places in the chloroplast. We can break photosynthesis down into two steps. The first one is the light-dependent reactions. It requires sunlight, which is why it's called that. And in the light-dependent reactions, you're going to use the energy from the sun. So use energy in light. You're going to take water, you're going to break it apart. We're going to use the electrons and the hydrogens in it. We're going to exhale the oxygen as a waste product. We don't need it right now, so we're just going to breathe it out, and it will help everyone else that needs to breathe oxygen. And we are going to take those hydrogens and those electrons, and we are going to use them in an electron transport chain again, and we're going to use ATP synthase again. And in the process, we are going to create energy molecules, ATP and NADPH. This one has a P, so not NADH. That was in the mitochondria. Now we're using NADPH. I think stick a P in there for photosynthesis. So those two energy molecules that we make during the light reactions will power the next step, which is called the Calvin cycle. The Calvin cycle is named for Melvin Calvin, who discovered it. He was a researcher at Berkeley. It's also called the dark reactions, but we're not using that term anymore because it doesn't have to happen in the dark. It also used to be called the light-independent reactions because it doesn't require light, hence the idea of dark reactions. You can use either of those names if you like, but most resources call it the Calvin cycle. In the Calvin cycle, we're taking the energy molecules that we created using sunlight, and we are going to fix them with an enzyme that's called Rubisco, and it's super important. Rubisco is going to fix carbon dioxide from the atmosphere, and we are going to take that carbon and we are going to alter it and alter it and alter it until we get a product called G3P. So G3P is what we make. Two G3Ps can be turned into a glucose. A lot of resources will say that the product of the Calvin cycle is glucose, and that's kind of skipping a step. It's totally true that this is going to be made to create glucose, but it's not glucose yet. So the product of Calvin cycle is G3P. I will call it glucose periodically just because it's simpler to think about, but in reality, there's an intermediary molecule. And then because this is a cycle, there's going to be a regeneration of your starting molecule, and then the entire thing can happen again and again and again, every time creating one of these molecules, which will be turned into glucose. That's the overall point of photosynthesis, is making sugar. Now, plants have the best of both worlds. They can make their own sugar, but then they still have to break it. So as soon as you create that glucose as a plant, you could add it to more glucoses and create starch or create cellulose, or you can take it to your mitochondria and you can break it doing cellular respiration, because plants still have mitochondria and they still need to break that sugar. So they make it and then they break it. Here's another schematic of what we're doing. In our shoots of our plant, we have our leaves, which are photosynthetic organs. Inside the leaf, we have these organelles. They're called chloroplasts. There's two membranes of the chloroplast. We are inside both of them. We are looking at the thylakoids, which are the sacs of phospholipid bilayer embedded in specialized proteins and enzymes. More than one thylakoid is called a granum. So this is a stack of thylakoids. This is a granum. We're going to be looking in the membrane in this. We're going to take sunlight. We're going to harness it. We're going to use it to split water. The oxygen will get exhaled. It's a waste product. The hydrogens and the electrons will be used to power the electron transport chain and make ATP. Those two products are ATP that we produced, and our high energy electrons in an electron carrier will go over to the Calvin cycle, which happens in the stroma. So we're in a different location now. Those will be added to our starting molecule, RUBP, which fixes with an enzyme called Rubisco. Rubisco takes carbon dioxide out of the atmosphere, turns it into an intermediary molecule. The number of G's and 3's and P's is unfortunate. I will not ask you to memorize the names, but I do want you to know that through the process of the cycle, we're creating G3P. That one I want you to know, G3P. G3P then can be turned into starches or used to create other components of your plant, such as other organelle tissues, fatty acids, sucrose, whatever your plant needs to be built, or can be used to break down and create ATP. Then the process regenerates RUBP and the entire thing can start again. Now let's go more detail. Here we are in the thylakoid. This is the thylakoid sac. So this is that little flat pita that we've been looking at. This is the thylakoid space that's inside the thylakoid sac. Here's your phospholipid bilayer. This is the stroma. This is outside the thylakoid, but inside the chloroplast. When we get our photosynthesis starting, we're going to take water and we are going to split water into oxygen gas, which is a waste product. It will just be exhale. No one needs it right now. And we are going to take the electrons from water to power the first protein in our electron transport chain. This first protein is called photosystem 2. So it was not discovered first. Photosystem 1 was discovered first. Photosystem 2 was discovered later. Even though photosystem 2 is used first, it's named photosystem 2. So in photosystem 2, you're going to take those electrons, you're going to put them into this reaction center in the center of your photosystem. All of these green dots over here are chlorophyll. When chlorophyll is hit by sunlight, it gets excited in a high energy state and it pops off little bits of energy. And those little bits of energy are used to power these electrons through the electron transport chain all the way through to the end. So we're going to use that energy to move electrons through their high state of energy. As the electrons move through these proteins in the electron transport chain, they're going to pump hydrogen against its concentration gradient. So the hydrogen concentration in the thylakoid space is higher than it is in the stroma. So we're pumping against the concentration gradient. That requires energy. Moving these electrons requires energy. That energy comes from sunlight. As we move those hydrogens in, we're creating that big concentration gradient. Your electrons continue to flow through the electron transport chain, and they kind of hit a low energy state right by photosystem 1. Photosystem 1 will activate by another photon. A photon is a particle of light. So light hits photosystem 1, it activates these chlorophylls that increases the energy level of those electrons. They can continue through the electron transport chain, going to the last protein, which is NADP reductase. Remember oil rig? So oxidation is loss, reduction is gain. Gain of what? Electrons. So if this is reductase, it's going to be adding electrons to something. And what is adding electrons to is the empty electron carrier. Here we're adding an electron. Now we have a full electron carrier. And we can take that high energy electron over to the Calvin cycle. We've also created that giant concentration gradient of hydrogen ions. Those hydrogen ions do not like to be crowded. We got some from water, we got some from the stroma. Either way, they want to go with their concentration gradient and they want to get out of that high concentration and go back to low concentration. So they're going to diffuse through our special enzyme, ATP synthase. And as they diffuse through ATP synthase rotates and it grabs a phosphate and it grabs a DP and it sticks them together, creating ATP. So ATP is created much in the same way as it is in the mitochondria. And that ATP is going to be used to power the Calvin cycle. So all those steps of the first half of photosynthesis, breaking water, using the electron transport chain, activating 2 photosystems, all of that was to create 2 high energy molecules that can be used to power the next step. Here's a visual of what these proteins actually look like. Each part has a name and we know its overall structure and we're still learning about the process. And it's just gorgeous and super complex, and I want you to see how beautiful it is. I love the way ATP synthase looks with its big turning head. You don't have to remember any of these parts of it, but I want you to see the complexity and the beauty that exists. Because when we draw it like this, it's cool, but this is not nearly as cool as knowing that the protein itself is this complex. I just love the intricacies of it. It's so, so neat. So that is the light reactions. Now let's get into the Calvin cycle. The Calvin cycle starts with an enzyme called Rubisco. That is a portmanteau. You just need to know this name. You don't have to know its entire full name. So Rubisco is the enzyme that is allowed to do carbon fixation. To fix carbon is to turn it from an inorganic form to an organic form. An organic compound has hydrogen and carbon. Carbon dioxide is just carbon and oxygen, so it's inorganic. To take this inorganic and add it to our beginning molecule, we are going to take it out of the atmosphere and attach it to a new molecule. And now it's going to contain hydrogen as well. So inorganic to organic. Rubisco, the carbon fixation enzyme. So Rubisco is going to fix carbon dioxide. It's going to create an intermediary. The starting molecule is RUBP. This is what Rubisco is fixing that carbon dioxide to. So this is a 5-carbon molecule. Carbon dioxide is 1 carbon. It's going to join together and then break apart. And our intermediary is 3 carbons because 5 and 1 is 6, and then break apart into 3. Now, we don't just start with one of these. We actually start with 3 carbon dioxide molecules and 5 of these, and everything gets a little more complicated, but we'll talk about that in a second. So carbon dioxide is fixed out of the atmosphere with the enzyme Rubisco added to the beginning molecule RUBP. We start with an intermediary molecule, which is going to be reduced. And then we get our end product, which is G3P. G3P is what we want. But we can't just stop there. It's a cycle. So we have to go through another intermediate stage until we regenerate RUBP. So we can break this down into 3 steps. Carbon fixation, taking carbon, adding it to RUBP using the enzyme Rubisco. Reduction, that's when we use the energy in our ATP and the NADPH that we created during the light reactions. And then regenerating RUBP so that the whole process can begin again. Here we have some numbers that will help us think about it. This is actually a really expensive process. We need 3 carbon dioxide molecules to start. We need 3 RUBP molecules. So that would be 15 carbons total, and then plus those 3 carbons, that's 8 carbons. Those, when they get added together with Rubisco, then split into 6 3-carbon molecules. So all the carbons there, it just has to rearrange. And now we're left with these 3-carbon molecules. These 3-carbon molecules need to be modified. It costs energy to modify them. So this is the reduction phase. In the reduction phase, we need 6 ATP. We need 6 NADPH. And all of that energy allows us to create 6 molecules of G3P. But we need 5 to regenerate RUBP. So we have to put 5 back into the process or the cycle breaks. And if the cycle breaks, then we can't create sugars, and that's a problem. So all of this energy and time just to get 1 molecule of G3P, that's our net takeaway. And 1 molecule of G3P is about half of a glucose. So we need to be doing this constantly everywhere in every single little chloroplast to be getting enough sugar for our plant to A, do its metabolism, keep itself alive, but B, create excess sugar so that it can build more body parts like cellulose and starch. If you look at wood, wood is just cellulose. So that's all sugary components put together. And that's a lot of energy investment and a lot of creation of glucose. Awesome. I have a video posted to Blackboard for you. It does a nice overview of photosynthesis. I think it's a really good video. And now I want to talk about a couple of misconceptions. So ask yourself, the process of carbon fixation is important because it converts The answer is inorganic carbon dioxide into an organic compound. That organic compound can be turned into a sugar. So that's what carbon fixation is. Fixation is inorganic to organic. The next misconception is where the carbon comes from. So the carbon dioxide used in carbon fixation comes from the answer is the air. It's carbon dioxide in the atmosphere. So all these plants behind me, all of that tissue, all of their body is made from carbon stuck into sugars that they pulled out of the air. So they're building materials from air, which is kind of hard to think about. So I have two videos up on YouTube for you to check out. Sorry, two videos up on Blackboard for you to check out about this misconception and how we're trying to fix it in the sciences. And then I want to show you a video right now about a photosynthetic experiment, and I'm going to give you some practice questions. So here's the video. What I have is a beaker. Here's water, and inside the beaker, there's a plant. This is a plant that has been cut. Its name is Elodea. They're going to turn the light on and turn the light off. Right now, the light is off. Nothing is happening. Turn the light on, and it starts to bubble. All of these little bubbles here are oxygen gas that are produced as a waste product of photosynthesis. So this little plant is doing photosynthesis, and we could count the number of bubbles to determine the rate of photosynthesis. When they turn the light off, you'll note that the bubbles stop. So you could see whether or not the plant is doing photosynthesis when the light is not on or when it is on. So that's the idea behind the bubble counting assay. Assay means how you set up an experiment. You could alter this. You could change the intensity of the light. You could change how far the light is from the plant. You could change the color of the light. But I want you to think about being able to count the oxygen bubbles as a way to measure the rate of photosynthesis. This always cracks me up. It's doing one giant bubble. Here's an experimental design practice question. I have the average number of bubbles given off in 30 seconds on different plant types. Elodea is what we were just looking at. It's an aquatic plant. These are two other types of aquatic plants. And then I have changed the color of the light, red, yellow, green, or blue. Now ask yourself, what is the question being answered? What would be a good positive and negative control group? And I want you to identify at least 5 variables to be held constant. So pause the video and do that, and I'll answer in just a sec. The question is, which color light is more effective for photosynthesis? A good positive control group would be natural light, maybe a grow light, or a full spectrum light. So like a light bulb that has all the colors. A good negative control group would be no light. Why? We want the positive control to give us a known outcome that will occur, and the negative control to give us a known outcome that will not occur. Identify at least 5 variables that need to be held constant. I would say that your water needs to be the same temperature, because temperature can affect photosynthetic rate. The distance of the light, the wattage of the lights, maybe even the brand of light bulbs. You would also want to control how much plant is in there. If I put like a whole bush of Elodea and just a tiny sprig of Utricularia, then that would be unfair because I would have more cells to do photosynthesis in the larger quantity. So anything that could mess up your results would need to be controlled, and those are your controlled variables. All right, imagine that you have discovered a new chemical. You have named it chemical X, and you think chemical X is an inhibitor of photosynthesis. To test this, you use the bubble counting assay with Elodea. So you've put Elodea in a beaker and you are counting the number of bubbles that are produced. What would be a good negative control? What's the independent and dependent variable? And what variables do you need to hold constant? Pause the video and ask yourself. A negative control would be where you know the results would give you a known outcome. So in this case, if I'm testing chemical X, the negative control would be no chemical X. So I would have just an elodea in the water, no chemical added to it. That would show me what photosynthesis looks like when it's uninhibited, and I could compare the other beakers to it. What are the independent and dependent variables? The independent variable is the thing you change. So that would be whether or not I've added chemical X to my plants. And the dependent variable is the thing that you measure. I'm counting the bubbles, so I'm measuring the photosynthetic rate. What variables do you need to hold constant? Temperature, light intensity, light distance, types of light. I didn't say anything about the light bulbs here, so I have to make sure I'm using the same light on all of them. The size of the plant, the size of the beaker, the time in which I count the bubbles, anything to make sure that my results are equal between each of my groups. As you review photosynthesis, a great way to study is to draw a chloroplast and label the specific parts, and then describe in words, either full sentences or say it out loud to your cat or your houseplants, what happens in each step of the light reactions and the Calvin cycle, and use as much vocab as you can. Identify every little bit of the chloroplast, identify the photosystems 1 and 2, identify that last protein, NADP reductase, ATP synthase, identify the enzymes that you start with in the Calvin cycle, and what molecule you begin and regenerate, and your end product. Note that it's G3P and not just glucose. Like as many details as you can when you say it or write it. I do recommend you spend time writing things. It does help you retain it, but talking out loud is important too. So tell someone how cool photosynthesis is. Another thing that would be great to do as you study is to draw a plant cell and label and list its organelles. The reason I add that is because cell structure and cell function is still really important. And we want to remember that a plant cell is not just chloroplasts. There's other components in there that are important to its function as well. And then the last thing that you might want to do is try a concept map. I've selected these vocab words for you to use in your concept map. They are tricky and kind of fun. I want you to see if you can add all of them, and note that there's things that you should add to the list that are missing. So after you do all these, look through your notes and figure out what is in there. Like, where does Rubisco fit in? So that might be a really fun way to study. Concept maps are great at identifying any gaps in knowledge. So that's what I always use when I study. Let me know if you have any questions. Email me anytime. My email is catherine.creech@mhcc.edu. And Max and Parker say hi and hope you have a great rest of your day. Bye.